Good afternoon, everyone. A very warm welcome to our third in our lunchtime seminar series. These are the fellowship lunchtime seminars, which we're planning to hold every term between weeks 2, 4, 6 and 8. And the whole point of these seminars is to bring together the entire community, past and present, to hear about some of the really exciting and interesting work that's being carried out by people on the fellowship. And so it gives me very great pleasure to welcome Lars Jansen, who is an associate professor and the Wellcome senior research fellow in the Department of Biochemistry. He's also the William R. Miller Fellow and Tutor in Biochemistry in the Hall. And Lars is one of our newest fellows who joined us last year and is now running the biochemistry for us in the college. If you look Lars up on Google Scholar, as many of us certainly did before this, you'll see Lars has got an incredibly impressive research publication already. And just before I began, I was saying to him, there are at least 3 papers that he's published in the past year, 2 of them in Nature, that I am about to go and search out. So it's very exciting to see the other research being done by people, both in all subjects across the hall. So very briefly, Lars's work, he'll tell me if I'm wrong, is focused around inheritance and how the cellular components are duplicated and passed on from one cell division to the next. And so I'm looking forward to now hearing all about this, as I hope all of us are. And he's going to talk to us today about genes, genetics and epigenetics. So Lars, over to you.
Thank you very much, Kathy. And I'm putting up my screen here. So it's a great pleasure to speak before you today. And in fact, especially it's a timely opportunity because as Kathy mentioned, I'm new to the hall, and so it's a nice opportunity to tell you a little bit about what we do and who I am and what I care about. And so I put up this title, Genes, Genetics and Epigenetics, and it's something that probably most of you are aware of and have thought about in some way or another, even though if you may not be natural scientists, but of course genes and genetics are common in sort of our social language when we talk about what we pass on to our children and the common traits that we have as human beings. And so I'll try to answer these 2 questions. First, what are genes and what is genetics really? And what do we think in modern terms what these things are? And then I'll come to epigenetics, which is sort of a perversion almost of what we typically consider genes and genetics. And so these are it's a sort of somewhat of an outlier that we put under this umbrella of epigenetics. And I'll try to explain what we mean by that. So the focus is on trying to answer these questions, and I'll try to weave in work that we do in my lab and put that in context with the literature and sort of the modern science that we work on today, but also put it into an historical context. So if we start at the beginning and sort of to get everybody on board on the principles here, when we think about genes and genetics, we think about inheritance. And what we almost intuitively, we all understand is that genes are somehow considered instructions or maybe even information that we carry within us and within ourselves that are important for development and from creation of a single cell zygote or an embryo to develop then into an adult individual. That's true for us humans, but that's true for almost any organism on the planet, including animals and plants. We require genes to give those instructions to develop tissues, organs, and ultimately whole individuals. Now, in terms of inheritance, what it is that eventually gets passed on to the next generation is whatever that is needed to recreate these individuals that we were ourselves. So we're passing on these essential instructions, and those is what we commonly call genes or genetic instructions. And so if we think about what is actually physically that is being passed on, this has been an ongoing debate for over a century. And the key cell types that are important in this context is the cells that we pass on to the offspring, which are our germ cells. So what I'm representing here are female oocyte and the main sperm cell that are together are the germ line, as we call it, or germ cells. So in some way, whatever genes are, whatever we consider them to be, in one way or another, they must pass through the germline, because that is, in the material sense, is what we pass on to our children, right? So that's important to bear in mind. So the germline is key, and then the question simply becomes, as a scientist, well, what is it in the germline that is being passed on, and what are those instructions? So, and I'll represent this in a different way, because so to go back a little bit into the origins of this concept. And what I'm showing you here is a diagram that is similar to the one on the previous slide, but I stole this and adapted this from this wonderful book by Jablonka and Lamb on evolutionary theory. And so what we're seeing here are the germline is represented by these little circles here. And that's what's being passed on from one generation to the next, generation 1, generation 2, and so forth. And so the genes is what's being passed on through those germ cells. But in our somatic tissue, that's written here, soma, which is basically our body, that includes our tissues and organs. What happens there is, as those genes are being read out, and those instructions are being put into place and executed in order to form an individual. So in sort of an historical sense, a lot of debate has focused on what it is that actually is being passed on, and to what extent can genes be influenced, for instance, in the context of evolution. So we know genes are important, but do we have actually any control over that, or does nature have control over those? And so for that, I'm going to take a step back and look a little bit at the history of science, and particularly in the context of evolutionary biology. So in the 19th century, in the 1800s, there were predominantly two views about how we thought that genes could change over time and across species. The first one was a thought of what we call evolution in a directed manner, where we have the ability of genes to be directly responsive to what happens to us in a particular environment. So individuals would be able to change their own genes in a way to create a better individual in their offspring. And this is a view that is by now has been largely attributed to this man, which was a French naturalist, Jean-Baptiste Lamarck, who was not the only one that had these ideas. He was the one that was a strong proponent of this type of a concept of evolution, but he was by no means the only person that thought this. Darwin, who is probably more famous, he had very similar ideas. And so the ideas in a very simplistic way, and this is, and let me just say this out loud right from the get-go, this is not what science actually looks like. So this is not what's happening, but this is something that was considered at the time. And so the idea of directed evolution, you can think of, for instance, examples that I list here. Imagine that you would use your muscles very frequently, like as a bodybuilder. Then if that would have an impact on your children, and your children would be stronger, or if you exercise your brain, your children would be smarter. So that would be wonderful, right? Except it's not true. It doesn't happen. But that would be a form of directed evolution. So in a way, if we put that in this context here, the idea would be that imagine that you have an individual that, due to some environmental cue or some environmental factor, happens to be an improved individual in evolutionary sense. Then there would be a way this arrow would essentially feed back into the germ cells and say, let's change the genes in order to maintain this state, so that across evolution and in subsequent generations, you would maintain this happy state, right? So this is a form of directed or Lamarckian evolution. So that was one school of thought. The other school of thought is that genes do not do this, and they simply change randomly. For the most part, genes don't change, and they're very stable, which is important because otherwise we would not survive as a species. But sometimes they change, but they don't do this in a directed way. They change randomly. But once they change in a way that is beneficial for a species, then the fittest of those are selected. So that's probably something that you're more familiar with, because that's the type of evolution for which we have most evidence today. So again, in this diagram, we have here these germ cells. And what happens is that the genes in those germ cells don't necessarily change in any particular way. They just simply produce individuals, they're somatic tissues. But at some point along the line, the germline changes. So genes within the germline, due to some environmental factor or just by random chance, they change. And this can happen, for instance, in a detrimental way. So most mutations in genes are bad for us, which means that a random change can lead to an unhappy phase, okay? And if this happens, the offspring of that individual would also be unhappy because the genes are changed. So this is, for instance, an example of this would be congenital disease, where you have a disease that is running in a family, for instance. But the converse can also happen, that you may have a happy mutation that somehow improves the species, and then selection will pick out those individuals, and this is how genes will spread to the population. So this is what we call Darwinian evolution, and in this case, very much the basic concept is that evolution is driven by chance. So this latter version is really how we think that genes are being passed on, and it became almost the evidence for that is so strong that it essentially became almost a dogma in science. And the one that sort of made it truly dogmatic, and I'm showing you one example here of an individual who was very powerful in this sense, a man called August Weismann, around the turn of the 20th century, so the beginning of the 20th century. He simply proposed that this division between the germline, which is the one that carries our genes, and the soma, which is our individual bodies, is so strict that there's a barrier between them, which is known as the Weismann barrier. So he said there's no communication whatsoever between these two, and therefore there is no Lamarckian evolution. Genes simply change randomly, and whatever happens to you as an individual has no bearing on your children. And he cited a famous example, which is a little bit of a ridiculous example, but it became famous because it's an example in extreme, which is, he said, if you would look at the breeding of laboratory animals like mice, and you breed a male and a female, you get offspring. If you would now do the same to animals in which you cut off their tails, the question then becomes, this is an acquired characteristic. These animals have acquired during their lifetime the loss of a tail. If it were true that this experience would have a bearing on children, that it means that the offspring of these animals would also have no tails. And of course, that experiment is a bit of a no-brainer, because you already know that that's not true, right? If imagine you would lose an arm and you would have children, your children will still have arms. We know that to be true, and we knew that to be true back then. So it's a bit of a nonsensical argument, but he made this argument in an extreme case. So that means that not everything we experience is passed on to the offspring, but doesn't mean that it excludes everything, right? Only in this particular case, it means that doesn't happen. Okay, so that's what happened sort of in the beginning of the 20th century in Western science. So Lamarckian evolution was discredited, and there is, in fact, even today in 2020, there is no evidence that genes can change in a directed way. However, in the Eastern side, and I put this slide in especially for Andrew Kahn, who is the organizer of this lecture series. And he knows a bit of the history of this. This is Tropy Lysenko, who was a very influential biologist and agricultural scientist in Russia in the 1930s. He was the main head scientist and the right hand of Stalin. And Lysenko was a great fan of Lamarckian inheritance, and he proposed that this was in fact happening, and he was very much interested in crop improvement. But the genetics was very highly politicized in Russia, and he prophesied the idea of Lamarckian directed evolution really without very much evidence. But the reason seems to be largely a political idea, because the idea that species can direct their own evolution fits very well with the Soviet idea that the proletariat can rise up, the people can rise up and improve themselves. The idea of Darwinian survival of the fittest was really viewed as a capitalist idea, that only the best can survive and everybody else perishes. So it became very political, and it really put back Russian genetics for 20 years, because essentially they were pursuing an idea that is turned out to be largely wrong. And Lysenko was essentially considered a charlatan in the end, and most of his experiments were never be able to be reproduced. So it's interesting how science sometimes gets caught up in politics, and this is very much so in the case of genetics. Okay, so let's go a little bit more concrete. So what is it actually that is being inherited, right? So we just sort of, it seems like genes are simply there in a stable manner, and they randomly change that drives evolution in the Darwinian sense. So we fast forward about to the 1950s with the discovery of a molecule that turned out to be extremely important for genetics and genes. And this is the 1950s in England, and the discovery of DNA, the molecule that you've all heard of, deoxyribonucleic acid, or in short, DNA. This is a molecular representation of that. This lady, Rosalind Franklin, generated data, and she was in London at the time, and she did experiments to demonstrate that DNA had a helical structure. And these two gentlemen used bits and pieces of information, including Franklin's data and some chemical intuition, to model a structure of the DNA that turned out to be largely correct. And that's what's represented here. Now, you all know this structure and this helical structure because it's part of statues across science campuses. And the reason why we all know this molecule and why it has been called the greatest discovery of the 20th century, at least in biology, is because it intuitively explains so much about genetics. For one, it carries information. It carries, and this you probably know, in its chemical backbone, there are part of this molecule instructions that are almost reading like letters. They're literally read like letters that are represented here. And those letters can be read by cells in our body to actually generate instructions to make tissues and organs. So DNA it explains genetics because it has means to carry instructions. It has a means to carry information. But secondarily, implicit in the structure, there is a means for duplication. And this is very important for a gene, because if you want to make a copy of yourself to create a child, right, if you have offspring, that means that there has to be some kind of duplication mechanism happening also at the molecular level. And DNA has this capacity because it's existing of these helical structures, these two strands that you see here, they can be separated. And each of them forms a template to make a new copy. And so this is beautifully encapsulated within the structure of DNA. So when that structure was discovered, all of these things became immediately clear. There was no evidence for it, but it was intuitive that that would work that way. And it turns out indeed it does. So DNA has a means to carry instructions and has a means to copy them. So this idea of the DNA being genes is so powerful that it almost became a dogmatic view in itself. And we became to view that DNA is the be-all and end-all of genes. The only thing we carry with us is genes in the form of DNA, and that's what we carry forward and nothing else. And this is the view that is being revised in modern science that I'd like to talk about a little bit more in detail. Now, if we think about our genome, this is a bookcase that you can find at the Wellcome Collection in London. The Wellcome Trust carries a art collection that is inspired by science and medicine. And there's an artist that literally printed out the entire human genome and put it in a bookcase. It's a tall case. So, I mean, if I would stand here, I would be reaching up to here somewhere. So it's a tall bookcase. The number 1 you can't even see in this photo. Then it's chromosome 2, 3, 4, and so forth. Each of these are chromosomes all the way to 22, and then we have the sex chromosomes. If you pick up a volume of this and you look inside, you see lots of letters here. And if you zoom in, these are pages and pages and pages of sequences of DNA. So only 4 letters. You can see the 4 letters here. So this is literally a printout of the human genome as we know it. Roughly 6 billion bases or letters. 6,000 million letters that make up our genome. And the technologies we have today to read these are so advanced, we can read an entire genome of an individual almost before coffee break in the morning. But however, reading and understanding these instructions are very different things, and we actually don't know what most of these letters mean. But they are essential for inheritance. So what it turns out is that these germ cells, the blue bits in here are the pieces where we have those DNA sequences, and they carry the genetic information. Now the question is that the only type of information that is carried forward? And I'm going to show you some examples that are violating this idea that DNA is the primary or is the carrier of all genetic information. So yes, DNA is important, but when you look closely, there's other things that come along with it. And so what I'm showing you here is a molecular view. This is, we're talking now at a scale of smaller than 10,000th of a millimeter, tiny molecules. And you can recognize here, all of you in the room can probably see, this is the helical structure that I showed you before. That's the DNA molecule. But in our cells, it comes attached to other things. These colorful things in the middle here are proteins. And they are sitting right next to the DNA. In fact, they're attached to it. and the DNA is an intimate relationship with these proteins. I recently wrote a blog for Teddy Hall, and I think it's still online, where I kind of made an analogy with a bowl of tagliatelle with meatballs. So it's a little bit like that. So the tagliatelle here is the DNA, and it's not this long helical structure that we see in textbooks. It's actually gobbled up and wrapped around these meatballs here. So it's, you know, our cells look a little bit more like this than what you would normally think of as a DNA molecule, right? So these meatballs turn out to be very important. So that would be the protein, and this is the DNA. The pasta here is the DNA. So if we think about the DNA, so again, the simple view is that this is long molecule of millions and millions of letters. But in our cells, the DNA is packaged with proteins, and so these little gray balls here are the proteins. So those are the meatballs in the analogy that I just used. Okay, and so now the question is, what do these proteins do, and what happens during inheritance? So if we look in the germline, and again, so we go from an adult tissue, our somatic tissues make germ cells, oocytes or sperm cells, that are essential for creating the next generation. And in fact, if you look in sperm cells, for instance, so we now focus on the male germline, what indeed happens is that during the formation of germ cells, all of these proteins, the meatballs, let's say, are being removed. We call that a step, is an erasure step. So it's really the DNA that is being packaged into this little cell here that creates the next generation. However, it turns out there's exception to this rule. What I'm showing you here is another version where some of these little protein balls are colored in red, which I just labeled them so that you can focus on them. And while it's true that most of these proteins are being removed in the germline in this erasure step, there are some proteins that stay behind. They stay on the DNA, they stay attached to the DNA, even in germ cells. So what that means in strict sense is that proteins are also inherited. When you have a germline where DNA is passed on, sometimes proteins are carried forward. Now, and it turns out these proteins are actually important. In this case, these proteins are important in the process of cell division. So what you're looking at here is an early embryo, not a human embryo, because those would be difficult to study. This is an embryo of a sand dollar, which is a marine animal that I just show you here for illustration. And you see that this embryo is becoming fuller and fuller of these little balls. These are the nuclei of cells. So these are essentially cells that carry DNA within them. And we're looking just at the DNA here. And whenever a cell splits in two, the DNA also gets copied because we carry the genetic information forward. And it turns out those little red proteins that I showed you before, those meatballs that stay attached to the DNA, they're important for this process of cell division. So here we have one cell, and then the cell becomes two cells in a process called mitosis or cell division. And these little red proteins that you see labeled here under the microscope, those are those red proteins that stay on the DNA in the germline. And they're important for in the next generation, in our offspring, to help cells divide and duplicate their chromosomes. So we have an example here where, in fact, in addition to the DNA, proteins are being passed on. Now, how does this work? And this gets a little bit to the nitty-gritty of the kinds of things that we do in my lab here at biochemistry, which is to ask, well, how can proteins actually make copies of themselves? How are they being inherited? So what you see here is a process of duplication, molecular duplication. So again, you have the black line here is the DNA. That's the helix that I showed you before. The proteins are these gray balls, which are the meatballs. And this is a large machine. It's also a protein that is able to make a copy of one DNA into two DNAs. So this is essentially what's at the heart of genetic inheritance, is that you have a DNA molecule that carries instructions that can be duplicated. Now, as you're doing this is a process of DNA duplication. As you're doing this, the proteins that are attached to the DNA, they're also being carried on over. So this used to be over here, these proteins, but now the DNA gets duplicated. And you see, these are four little meatballs. Two of them go here, two of them go here. So the DNA brings along the proteins and they stay attached to the DNA. So if the DNA carries instructions, the proteins can also do that and they stay on the DNA to be inherited. Okay, so at the end of this, you have two molecules of DNA, which will become two cells or two individuals that still have those proteins attached to them. But there's a problem here. And the problem is that we started with 4 proteins, but after division, we only have 2 because we need 2 for one DNA molecule and 2 for the other. So at some point, even though we're inheriting DNA here, this is called protein inheritance, we still need to make copies of them. And this requires specialized machines. So, and this is really this is the nitty-gritty of the kinds of things that we're studying in the lab, is what is the machinery in our cells that is able to do that? So there's proteins that are involved in reading. It's almost like reading and writing, reading a book and writing text. So you read the instructions, which is the presence of this red protein here, and it's able to recognize that and put in a new protein. So the new protein I label in green here, so you can tell the difference. So we copy proteins into their position. So in the end, we have these old proteins that were instructive, and they make a new protein next to it. So in the end, we have 2 molecules and we have a full set of proteins. So this is to show that proteins are also being carried forward, and this is a form of epigenetic inheritance. Now, this sounds very detailed and esoteric. So let me take a step back and give you some other examples, okay? So here, what about genes? So I told you about these proteins that are important for cell division. But what about all these genes that we have, right? So what I'm showing you here is a long DNA molecule, and these little blocks are units of DNA, which we call genes. And in humans, we have about, give or take, 25,000 to about 50,000 of these genes, depending on how you count them. So I gave an average of 40,000. We don't all agree necessarily on where a gene begins and where a gene ends, but there's many, many of them. So we have tens of thousands of these genes. And typically these genes are involved in making components within our cells and our bodies. So if I give you an example, for instance, we can consider the genes as little switches. If a gene is turned off, it means it doesn't do anything. But if a gene is turned on, it means it's producing a component like a building block. So if you think about a house, for instance, you imagine you have a gene for laying the bricks, and you have a gene to make a window, or you have a gene to put a roof on the house. And collectively, you build a house from that set of instructions. In this case, we have a set of genes where this one is on and that one's on, that will, for instance, make muscle cells. And so each gene is like a molecular switch. It's turned on or it's turned off. And by switching on different genes in different cells, we can make different cells. So for instance, in this cell type, these are blood cells, one gene is maintained on, but some other genes are turned off and others are turned on. So this one used to be off, now it's turned on. This one used to be on, but now it's turned off. And that one's turned on, which wasn't on before. And the same thing over and over again in different tissues. This is a neuron where all of these are off, but that one's on, okay? So you can easily imagine with tens of thousands of genes, you can make enormous amounts of combinations of switching genes on or off. Now, what does this have to do with inheritance? This is what we call development, right? You can make from a single embryo, can make an individual with lots of different kinds of tissues. Now, it turns out that the switch to turn genes on or off also has repercussions for those proteins that are sitting on the DNA. So if you think about a gene that's off and a gene that's on, now we zoom in a little bit more closer. You see the DNA, the black line, but we're back at the meatballs level, right? You can see those proteins attached to DNA. And here we see that the proteins are different when genes are on or off. And this creates an opportunity for inheritance. So the question now becomes, is the position of those switches, can those be inherited? And the answer is sometimes yes. And that's very interesting. So I'll give you a couple of examples from different kind of kingdoms of life. This is a plant, and plants are very good at doing this trickery that I'm going to tell you about. So this is a plant called the toadflax. It's a very, very common plant, even in this parts of the world. And a typical morphology of this plant is has this kind of flower shape. So it's a flowering plant. And in this case, this flower will develop if this particular gene is activated. So let's say this gene is on, you get this flower type. Now, in some cases, if you get a mutation in a gene or some other way that this gene is off, what happens now is that you get a very different flowering plant. So the plant is still alive, but the flower structure is very different. So when the gene is on, you get this, but when the gene is off, you get this. And it turns out that this can even happen when the plants are genetically identical. So the DNA is the same, but they activate the genes differently, and so they got different morphological structures. And what's really striking in this case is that ability is maintained across generations. So in these plants, when a gene is off, when the gene is on and forms a structure, their offspring will maintain that flower structure. When the gene is off and you form a different flower structure, their offspring is also maintained. And these can go on for tens of generations. So originally, these plants were actually classified as different species. It turns out they're genetically identical, but they're maintained in a way where the switch gets stuck in a way. And so the switch stays on across multiple generations, or the switch stays off across multiple generations. And this happens not only in plants, it can also happen in animals. So this is a famous example in mice, for instance. This is a gene called agouti. If this gene is turned off, then the mice develop with a brown fur, which is the typical wild-type mouse, as we call it. That is the normal mouse that you would see in the wild or also in laboratories. But if this gene is for some reason turned on, which is usually not the case, what you have is you get a yellow furred colored animal, and they also suffer from diabetes and have other problems. So this is a well-known gene in mouse genetics. But it turns out that although not as extreme as in the plant case, also this off or on state is inherited across generations. So you have a brown mouse that is in a agouti, let's say, brown state where the gene is off. A proportion of offspring will be brown again. Some of them will be sort of mixed, and about 40% are yellow colored. But if you start with a yellow mouse, then the offspring is never brown, even though these are genetically identical animals. So fur color in this sense is, at least for a couple of generations, propagated depending on what was the fur color of the parent, not because of the genes that are within those animals. And this can even be influenced by diet. So if you feed these animals certain types of food, you can kind of push the ratio towards brownness or towards yellowness. So heritability in this case is influenced by diet. Okay, that doesn't mean that everything is inherited. You know, when you cut off a tail, that will not be inherited, but there are certain properties that are inherited, both in plants and animals. Another example here, and this is a final example before I move on to the last bit, is this wonderful example of a water flea, which develops differently depending on whether it's in the presence of predators. If a predator is present, this animal, which lives in small ponds, so this is a microscopic animal, develops these morphological traits which helps it to evade the attack by this predator by using these spines here that prevents it from being eaten. But interestingly, if this animal that was experiencing the presence of the predator, you remove the predator and generate offspring, these are the children or grandchildren of that animal, they also develop these structures even though they never saw the predator. So this seems to be what is called an adaptive change. So it's sometimes those changes seem to be beneficial. And it's almost like these animals trying to predict the future, thinking, well, if I was living in the presence of predators, then maybe it would be a good idea for my offspring to also be protected against those, right? So it's almost like what we call this Lamarckian principle, but not because genes are changed. We cannot control those genes, but it seems like whether the switches are on or off can be controlled. Okay, so far what I told you is talked to you about this bit down here, where we go from one generation to another through the germline. And I told you that DNA is not the only thing that gets passed on, but sometimes also proteins. And we're interested in the mechanisms of how this works. Now, in the last few minutes, I want to switch to another form of inheritance that we work on in the laboratory that I think is really fascinating. And that has to do with passing on genetic information and epigenetic information within cells in our body. So now we're not going between generations, but we are moving within a generation as we are developing from an embryo to a somatic individual. And of course, this involves many, many cell divisions. I show you a movie here of cells that are in the dish. This is something that a movie that came from my laboratory, where you can see these are simple individual cells sitting in a dish, and we're filming them dividing under the microscope. These are cancer cells, so they grow really, really fast. This is a movie that is running over a couple of days. And you see that the cells become over time more numerous. And that's because they're dividing. And every time they divide, they copy their genetic information, their DNA, but they also pass on other potential pieces of information that we want to find out about. So I'm going to give you an example of a heritable form of information that is not encoded in the genes that seems to be very important for human health. And this we call cellular inheritance. So we don't inherit across generations, but within ourselves, within our body. And we stumbled a couple of years ago on a peculiar phenomenon. And I call it here peculiar phenomenon, because we still don't quite understand how it works. And this has to do with innate immunity. So our innate immune system is an immediate response system, like a policing system against invading pathogens. And these involve cells like macrophages you might have heard of. And so what these cells do, they find viruses and bacteria and directly attack them. It's called innate immunity. So this is not the type of immunity that you would have, for instance, with antibodies, where you have lifelong protection because you had a vaccine early in life that protects you because you generated antibodies, which is what we're trying to do with the COVID vaccine, for instance. This is an immediate response, but it turns out that also innate immunity has a form of memory. So I'm showing this kind of graph here. This is time. Imagine you have an individual with immune cells, and at some point in time, you challenge this individual with an immune challenge, like an infection. You get infected with a bacteria or virus. Then these cells give an immune response. Now, it was recently discovered that the strength of this response depends on whether these cells are seeing the pathogen for the first time or whether they've seen it before. And this is called priming. So what happens here is if you have somebody, let's say an individual or an animal in the laboratory that was initially exposed to an infection, and then later in life is exposed again, the response to that infection is much stronger. So there seems to be some kind of memory effect where this individual, a patient or an animal, remembers the prior infection. And in this case, has nothing to do with the well-known memory that we have in the adaptive immune system. It's something else. It relates to gene expression. So I'll just give you two examples here. For instance, in mice, if you treat mice with an infection, such as a herpes or a cytomegalovirus infection, then those animals, they become more resistant to infections later on. Let's say 3 months later in life, if you challenge them with something completely different, like a bacterium, they're more resistant to that bacterium because they were challenged before by this viral infection. So it's a broad innate immunity against multiple infections that is memorized within their tissues. Also, in humans, there is evidence where, for instance, if you have vaccination against Mycobacterium tuberculosis, which is a widely used vaccination program, BCG, then there is evidence that in children, even within 12 months after the vaccination, they are more resistant to other types of infections. So there seems to be a molecular memory present in these individuals. And so what we're trying to do in the lab is to understand the molecular basis for that. And to do this, we've simplified the problem. What we're trying to do is we move away from human patients or animals. We look at individual cells, and we're looking at what genes are regulated in those cells by those in the innate immune system. And what happens in the innate immune system is these cells that attack bacteria or viruses, they make small molecules called cytokines. And cytokines, they activate genes. So let's call them immune activators to keep it simple, okay? So what immune activators do is they activate the immune response, but really what they do is they activate genes. So we're back to this molecular switches. Remember the switch on and off that I told you about before? Now, we're trying to understand whether this innate immune memory that we're seeing in patients and animals, whether that has any relationship to the way that genes are memorized within these cells. So what we do here is we basically take cells in the lab, we stimulate them with this immune stimulation and look at genes that are activated. And it turns out that some genes, when you activate them the first time, they become activated. You see that with this little bump here. But then you remove the stimulus, those genes are switched off again. So you switch on the gene, then you switch the gene off. But then much later, when you add a second stimulus, those genes are being activated much more strongly, just like the immune system in the patients. So this is what's called trained immunity, and we're trying to understand the molecular nature of that. So what we're assuming here, and we have evidence for that, there's immunity genes in our genome that are normally turned off. So again, we have lots of genes. We're focusing on one here. It's turned off, and let's just focus on that gene. And we follow this graph here. So we have a cell in a dish. The gene is turned off. We activate the gene with an immune stimulator, which is called a cytokine. And now the gene is turned on. We switch on the gene. So it's like this, we flip the switch by activating the gene. But then over time, even for many days or sometimes weeks in culture, these cells continue to divide and they continue to pass on their genetic or whatever information they have to their daughter cells. And then weeks later, if we add the second stimulus, they remember that they were activated because now we have a much stronger response. So there is some kind of epigenetic state. There is a memory within these cells that remembers that they were exposed to this immune response before. And it looks like we're not looking at a switch here, but it's almost like the dimmer that you have in your living room, where you can turn on the light, you know, a little bit lower in the evening or brighter during dinner time. And it's the same thing happening here. We have a small response, but then this response becomes much bigger. So how is gene activity remembered? It's a little bit like this transgenerational effect that I showed you before in the plants and animals, but now within cells. So I'll close with the evidence that we have so far, which is it's a little bit nitty gritty, but I'm trying to explain the concept. It turns out that what we did is we tried to see how many genes are behaving like this by simply looking at all the genes in the genome. And it turns out that most genes that behave like this, which have this memory, they're all sitting next to each other. So they are in groups of genes, and that probably is important. And it turns out that, so there's not one gene that is doing this, but there's multiple, and there always comes in groups. And it turns out that the reason why they're in groups is because they're held together in some kind of arrangement like this. There's a little lock on them that keeps these genes turned off. So let's go back to our experiment. And I think it's the last experiment I'll show you, because it might be a little bit confusing, but I'll walk you through the experiment. So it turns out that when cells have never been exposed to an immune stimulus before, those genes that are important for attacking the virus or the bacteria, they are locked up in this way. So they're turned off, but on top of being turned off, they're somehow packaged in this way inside our cells, inside the nuclei. When we activate the genes, we turn on these genes and the lock is broken. But now in the intervening time, even through many cell divisions, the genes are turned off. But in some way, they've lost this locked arrangement, which means that when they're exposed again to an immune response, they respond much faster and quicker. So we think that this is possibly a molecular mechanism for how cells can transiently, for a number of cell divisions, for sometimes weeks or months in an individual, remember exposure to pathogens. And that might be important for our capacity to turn on an innate immune system. But from a molecular standpoint, it turns out, again, that genes can be turned off, but their activity is remembered even through multiple cell divisions. Okay, so I'm going to conclude now and just summarize what I told you. So I show you this diagram here. Genes are being inherited from one generation to the next. I told you that in addition to DNA that we know of, we have proteins that are being passed on. And that has functional implications, not only proteins, but other molecules that I didn't talk about. So sometimes more than DNA is inherited. The other take-home message is that in our tissues, in our individual bodies, there is also memory of earlier exposure. In this case, we're using immune exposure, but it's also true for other types of signaling within our bodies. And I show you an example of innate immunity. Thirdly, I showed you some examples of how the somatic tissues can somehow feed back to the germline. Not because we're changing genes, but what's being inherited sometimes is the switches that turn genes on or off. And it's really at the frontiers of science to figure out to what extent this is happening, whether this is very common or whether this is relatively rare and limited to the examples I gave you. So I'll close with going full circle. And I told you about these initial ideas in the 19th century of how genes might change and how we influence them. And we have this directed form of evolution, where genes are directed in a way that would be beneficial to us, which has been disproven, which is this Lamarckian idea. And Lamarck is sort of a persona non grata now in biological sciences because it turns out to be all Darwinian. But I told you that there are pseudo-Lamarckian effects where genes are not changed, but the way that they're switched, they are changed. However, I should say, and this is my last note, is that this reality has now changed. The ability to change genes directly in our germline is in fact not happening in nature, but we as humans with our human brains have now been able to achieve this. And to the extent, and this is probably and you're aware of this, and it's all over the news, and there's even Netflix TV series about this. And of course, I'm talking about CRISPR, because we have now gained the ability to actually directly change genes at will, even in a germline, even in humans. And this is a very current technology. And in fact, these two ladies just a couple of weeks ago shared the Nobel Prize for discovering this technology. And so in fact, Lamarckian evolution is something that, even though not happening, is in fact something that we could do. And this is of course an exciting but also a scary prospect of, you know, how should we move forward with this type of technology that might change our own future fate. So I'll leave you with that note and take any questions.
Thank you very much, Lars. I'll clap on behalf of everybody to say thank you for an absolutely fascinating talk.